Nuclear imaging technology used to detect necrosis, i.e. dead tissue, currently lacks the precision to avoid misdiagnosis, which occurs roughly 12% of the time. Now, the presence or absence of necrotic tissue has surgical, medical, and total care implications. Fortunately, Dr. Awasi and his research team have invented a novel kit which converts the most commonly available PET imaging agent, FDG, into fluoroglucaric acid, FGA. When administered to a patient, FGA will bind to the totality of necrotic tissue, and its radioisotope F18 will be illuminated by PET scan, providing the highest resolution image available. Our interdisciplinary team has conducted no less than 25 interviews with experts in the fields of radiology, cardiology, neurology, oncology, and a variety of other ologies. What we determined is that FGA has the potential to improve patient outcomes in 4 distinct use cases: myocardial infarction, i.e. heart attack, stroke, cancer, and critical limb ischemia. Furthermore, we've identified the best commercialization strategy to bring FGA from the bench to the bedside. Creating an academic spin-off can produce a return on investment of 327%, a payout to the university of $75 million, and as a platform technology, FGA can generate upwards of $1.9 billion in revenue over the course of 10 years on the market. Thank you all for attending today. My name is Dan Sloat. I'm a third-year JD MBA.
My name is Reagan Moulding, and I'm a second-year occupational therapy candidate.
My name is Rachel Davis, and I'm a first-year MD candidate.
So to better understand this revenue opportunity, we first must address diagnostic imaging and the current shortfalls with identifying necrotic tissue. Some of the most common imaging modalities are MRI, CT, SPECT, and PET. You've probably heard these terms at your doctor's office, or perhaps you've had a scan yourself. MRI and CT are both structural imaging modalities, and they are used to identify bodily structures such as bones. Whereas SPECT and PET are both functional imaging modalities, and they are used to image physiological processes occurring within the body, such as blood flow or hypermetabolic tissue. Today, our focus will be on a novel PET imaging agent and its ability to bind to necrotic tissue. Because our team has found that currently on the market, there is not an imaging agent that has high sensitivity and binds to necrotic tissue. And this lack of technology provides a wide-ranging implications on patient care. For example, one of the methods of tracking cancer treatment is by using an imaging agent that binds to hypermetabolic tissue. Means it is binding to the viable cancerous tumor. Therefore, the physician then assumes that the area around it that is not lit up is necrotic. However, this process is lacking in diagnostic accuracy and can have a sensitivity and specificity as low as 43%. Secondly, some of the current methods of diagnosing myocardial infarction or heart attack do not specifically pinpoint whether there is the presence or absence of necrotic tissue. And therefore, 1 in 9 people are told that they didn't have a heart attack when in fact they did. And lastly, after talking to Dr. Ringus, who is an orthopedic surgeon in Norman, our team learned that there is not a standardized assessment used to identify exactly where necrotic tissue ends and living tissue begins before amputation. But fortunately here at OUHSC, Dr. Awasi and his lab have a solution. His lab has created a kit that forms fluoroglucaric acid, or FGA. And FGA is the first PET imaging agent that binds to necrotic tissue. And it has a high sensitivity, so that physicians can be confident and certain that they are seeing the necrotic tissue in its entirety. And this will decrease the chance of a misdiagnosis, provide a standardized way of tracking cancer treatment, as well as provide a uniform method to ensure amputation accuracy. So like I mentioned before, this begins as a kit. And this kit is non-radioactive and therefore can be stored on the shelves of the nuclear pharmacy for up to 6 months. However, whenever an FGA scan is needed, the reactive components are then added. And it's important to note that one of these reactive components is F18-FDG, which is the most common imaging agent on the market for PET scans. Therefore, it is readily accessible at a nuclear pharmacy, which is where this reaction would take place. The reaction time, it takes roughly 5 minutes, and then FGA is created. From there, it is sent to the imaging facility and injected into the patient. The imaging agent then binds to the area of necrosis, and that's what will light up on the PET scan. And here we have images to showcase just how accurate FGA is. We have an FGA PET scan and a SPECT scan, and these are images of brain slices from rat models with induced stroke. And as you can see, the lit up area in the FGA PET scan correlates exactly with the white and necrotic tissue in the brain slice. However, when we compare these two images to the SPECT image, the imaging agent used for SPECT is targeting that hypermetabolic tissue, and therefore the darkened area is assumed to be necrotic. But as we can tell, this process does not provide the exact boundaries of the necrotic tissue as compared to FGA. And we asked Dr. Baldwin, who is a nuclear medicine professor here at the university, as well as Dr. Brannon, who is a radiologist at OU Children's, to compare the two modalities of PET and SPECT, and both of them said, hands down, PET is significantly better. So now that we have the most superior modality, when you compare FGA to other imaging agents on the market, it's clear that it is also the best choice, because it's the only one that targets that necrotic tissue, but also has a high resolution and sensitivity. So currently, Dr. Awasi and his lab are in the preclinical trial space. They have tested FGA in rat models with induced myocardial infarction and stroke. They have filed a provisional patent on this technology, and his lab is currently investigating the mechanism of action. So now that we have a better understanding of diagnostic imaging and the role FGA can play, Rachel's going to tell us about the types of patients who will benefit most from it.
All right, thanks, Reagan. So as Reagan mentioned, we've got a great product here. So what exactly do we do with it? So our team throughout the past semester has looked at about 8 or different 9 potential use cases for FGA, and has narrowed it down to 4 specific cases where we see FGA can have the most clinical impact. Those 4 are here: myocardial infarction, ischemic stroke, critical limb ischemia, and oncology. Together, they make up a $163 billion market in the United States. So as we looked through these different cases, we recommend that first we start in oncology and then branch out into the other 3 use cases. And that's for a variety of reasons. The first is, as you can see, the numbers are just larger for oncology. There is a larger market, both in the number of patients that develop cancer each year and in just the total diagnostic market size. In addition, PET scans are kind of synonymous with cancer treatment currently. Almost every single cancer patient will have an initial PET scan for staging, and then recurrent PET scans throughout the time of their treatment. And that connection between oncology and PET scans leads insurance companies to be most likely to reimburse a physician or a clinic for an FGA scan because it's such a common modality used currently. In addition, and Dana will get into this a little more later, we are looking at oncology not as a purely diagnostic use of FGA, but rather to track the progression of treatment over however long that treatment takes. That could be chemo, radiation, any kind of oncological treatment. So these would be recurring scans, not just a one-time transaction. So diving a little further into this $124 billion market that is oncology, we have identified 3 specific sections where FGA can have the largest clinical impact and that we recommend the team pursue going forward. Those 3 are genitourinary cancers, brain cancers, and liver cancers. And the reason for that is because of the difficulty in diagnostics and especially imaging in those particular areas. When you look at genitourinary cancer, which includes cancers of both the male and female reproductive organs, as well as bladder and kidney cancer, you see there can be quite a variety in the way that those structures are anatomically put together. There's a lot of variety throughout different humans, which can create challenges for a surgeon or an interventional radiologist who's wanting to go in and take a biopsy. In addition, you have to worry about maybe obesity or some other issues that you might see in patients, which makes those structures particularly difficult to get to, which makes both biopsies and potential surgeries invasive, risky, and sometimes just overall dangerous for the particular patient. If you can replace those dangerous procedures with a non-invasive FGA scan to determine exactly where the extent of tumor necrosis is, you could prevent some of those risky surgeries, which would in turn save both time, money, and risk for the patient. When we look at brain and liver cancers, those are areas where the current modality FDG is very commonly taken up. because FDG binds to hypermetabolic tissue, and there's a lot of things going on in your liver and brain on a normal day-to-day basis. It can create a lot of misdiagnosis and problems for the radiologist to determine if there really are maybe a new met on a liver, or whether it's actually just an old tumor or an old metastasis that has been necrosed. So the addition of FGA to the kind of a oncologist toolkit, if you will give them much greater accuracy in making their clinical decisions. Going forward, we recommend that the commercialization start first in the North American market and then expand into potentially Europe or Asia. And this is a recommendation that OTD specifically asked for. We have secured a provisional patent in the United States and are working to keep that secure. However, we see that there are large markets, particularly in the EU, and regulatory schemes are relatively friendly there, so that could be a great place for FGA to expand in the coming years. And so now that we've seen who exactly can benefit from FGA and the markets that we're looking at, I'm going to turn it over to Dan to talk about how we're going to get there.
Thank you, Rachel. Now, a key takeaway today is that all players in this value chain can profit. According to Michael Shade, an IP attorney at Dunlap Cotting, the above represents a typical licensing arrangement for an academic spin-off onto market. So with a spin-off, we're recommending the name Zolatil Radiopharmaceuticals. And I'd be happy to elaborate more on that name during the Q&A. The spin-off will be responsible for raising funds, conducting clinical trials, establishing a minimum viable product, and finding a suitable industry partner for acquisition. We're proposing Siemens as our target acquirer because they have global distribution. At that point, once Siemens manufactures and sells the FGA kit, it will go on to end users such as Stevenson Cancer Center. It's worth noting that the FGA kit, when combined with 1 dose of FDG, will produce 2 doses of FGA. So while we're pricing it from a unit economic standpoint at $300, the cost to an individual customer will be just $150. As I said with Siemens, you can see on the left, along with Philips and GE, collectively they dominate the FDG market. And on the right, you see PetNet Solutions, Cardinal Health, and Midwest Medical Isotopes. These are regional manufacturers who all service the Oklahoma City area. What we've determined is that PetNet Solutions, as a subsidiary of Siemens, could be an excellent strategic partner. Because as we move forward through development, we hope that we can organically grow into that global distribution. Now, in the alternative, Cardinal Health, we can achieve at least national distribution. And then finally, Midwest Medical Isotopes, which is led by Dr. George Chaco. He's an alum of OU and has already agreed to provide favorable terms to the university, at least through development. So again, FGA kits can produce upwards of $1.9 billion in revenue over the course of 10 years in the market. Few of the assumptions involved. Industry reports indicated that R&D could cost upwards of $100 million. We have 2028 as the target acquisition date, and we've estimated roughly $500 million for that valuation. Now, according to Dr. Rick Alvarez, CFO of Tetherex, he explained that allowing the spin-off management team to retain 85% equity will properly incentivize them to ensure commercialization. Still, the university retaining 15%, they'll achieve a $75 million payout. We started with 10% market penetration in oncology, and you see a waterfall effect into CLI, stroke, and finally heart attack. In the initial year, we estimate that FGA can bring in a revenue of $72 million and can positively impact 160,000 patients. In its final year of patent protection, we estimate that it can bring in $340 million and improve the lives of 1.2 million patients. I'll turn it over to Rachel to discuss next steps.
All right, so as we've kind of alluded to, and as Dan mentioned, this is going to be a very long process to get FDA from where it is now to where it will be, in 10, 20 years affecting patients. That being said, there are some key steps that can be taken in the next year or so to really jumpstart that process and get FDA on its way to helping the patients that need it. The first thing on the R&D side would be to determine the mechanism of action of FGA. It's my understanding that this is a research interest of Dr. Wassing and his lab, and determining that exactly will be incredibly helpful as we go through these FDA trials. In addition, since we are looking at going into oncology first, it would be a good idea to go ahead and start the preclinical trials for oncological use as well, so that we can leverage that into those initial FDA trials. On the regulatory and IP side, the major thing will be solidifying that patent protection in the United States and looking into what needs to be done to secure patent protection in the EU through the EMA. On the business side, two major things. The first would be establishing a management team. And I will note that that is not just a business management team. This also needs to include identifying clinical collaborators that could come alongside Dr. Wassing's lab and work with them throughout the different phases of clinical trials. And finally, the last thing that needs to happen in these first couple of years is simply raising the funds to get us through clinical trials. That can come from a variety of places, whether that be national or state grants. It can come from investors, or with the NIH as well has some different opportunities there. And so overall, throughout this entire semester, we have conducted 25, almost 30 different interviews with physicians and experts in all kinds of fields. As we mentioned before, we've narrowed it down to 4 major use cases where we see FGA has the ability to make a very large impact, not just monetarily, but in the clinic for specific patients. Going forward, we see that this can have upwards of a $75 million payout just for the University of Oklahoma alone, and that we can impact around 5.9 million patients throughout those first 10 years on the market. So I will say for myself and for our team, it's been a joy and a privilege to work on this project this semester. I know we've all learned quite a lot. So at this time, I'd like to conclude our presentation, and we'll take any questions you might have.